Okay, so well, I am going to talk now about spectral phasor for hyperspectral imaging. And the first time that I give this talk in at the LFD, Enrico put me this on front and saying, well, what is in common and how we can take advantage of the phasor to also acquire and analyze imaging from hyperspectral imaging. And I was uh, um, working on this even before I moved to the LSD. And the reason why I was working with this is because when I was doing my thesis in Uruguay, we didn't have any film microscope in Uruguay, neither a film machine for cuvette experiment, but we got a hyperspectral microscope at the Instituto Pasteur at that time. And I remember once we met with Enrico on a conference, and he said, well, Leonel, now we are able to do phasors in Uruguay. And I said, no, Enrico, no way. We don't have any machine to do phasors. And Enrico sit with me and explain me how to do phasor on hyperspectral imaging. And after that, it was my conversion to the phasors and hyperspectral imaging and film imaging when I joined the LFT. Okay, so we already have learned about how the phasors work for lifetime imaging, and sorry for that. And what we learn is, well, we will have basically you see, well, components with different lifetime depending on if they are short in blue or larger in the green, and the modulation and phase change depending, of the lifetime. And the thing is, well, what we can compare in between hyperspectral imaging and lifetime? Well, first of all, as Enrico mentioned, and also Richard and Dave comment today, for do the phasor transformation, we need a period. And this is important thing. We always need to have a period, and this period need to be fixed. We cannot change, because otherwise our reference transformation will change, right? So in the hyperspectral imaging, we have a period because our detector has a period by default. It starts at a point and finish at a certain other point. And we also have got a function that can be, fluorescent with a certain maximum and certain bandwidth that can be transformed in our phasor as a point with a certain modulation and a certain phase. And this transformation will capture basically the spectral shift as well as the broadening of the spectra by changing the phase, which refers to the maximum or the center of mass of the spectra, and the modulation will capture how broad or shrink is our spectra. And I will show in detail how this works and how all the transformation. But if we compare the two, we can immediately learn that, yes, of course, we can do phasors on hyperspectral imaging. And we will see how this can help and how we can use this to take advantage of this information. But before to go to the phasor, what I would like to highlight is the enormous amount of technology around that we have today to do hyperspectral imaging. So almost every brand that we can buy today, Zeiss, Leica, Nikon, Olympus, offer hyperspectral imaging. And how most of them do this hyperspectral imaging? Well, we have an image with X and Y resolution. And then we got a third dimension, which is the lambda. And this lambda can be captured by different way of different array. Well, we can have basically an optic device that will open the fluorescent, right? So this can be a grating or a prism that will discriminate between the all colors. And then we can have either a single detector or an array, and we can collect point by point or all the points together in a single shot. This is the case, for example, of Zeiss, and also the case of Nikon. By the case of Leica and Olympus, they got step by step the whole spectrum. And of course, everyone has advantage and disadvantage. Time, exposure of your sample, etc. The thing is, there are some other solution for high spectral imaging, and one of them are basically the camera. If you got a camera and you project a point in your camera, well, the spectra in your camera, you can collect the intensity in a space in the chip of your camera, and you can assign, if you calibrate, what is the wavelength of each of the pixel, and you will have really great resolution on the nanometer resolution. But nowadays there are some other approximation, and I would like to highlight this approximation of a sine and cosine transmission shape filters, and I will go in detail at the end of talk, that we can capture the spectral information without obtaining the spectra, just doing the phasor transformation on the acquisition of the data, and we will discuss this in later on. So basically, in the size, we collect a stack of 32 images, and you can see how it changed the intensity. So you can, looking at this, you can see that there is a spectra with some maximum around where I have the pointer now. And then most of the time, the companies offer, a pseudo color image where they assign to each of the channels the color for the maximum that you got in your example. Okay. Every time that we have something to admit, it pop up in my screen. So, well, what is the classical way that people use the phasor? Well, the classical way that people use the phasor is basically by the hyperspectral imaging is basically you collect the spectra, and then what you do with the information, because you have at least 32 channels, right? So most of the time, in the companies that sell the instrument, they offer different kind of methods like supervised classification analysis, principal component analysis, and linear mixing. And in all of that, they go in different directions to achieve that goal. But basically, if you collect a spectra and you have an expectation for one spectra here in red or another spectra here in green, you can deconvolute the contribution of one and over the other, and you can assign the amount of one component or the other pixel by pixel. And at the end, you can split the color that you have in the image. And this can be for 2 channels, for 2 colors or 3 colors, etc. So far, what you need is a model. You need to know what you have in your sample, and you need to know what is the maximum, what is the full width at maximum of your sample, in order to introduce this information in the convolution and give the information to the software to acquire. But the problem is when we want to study samples where the spectra is not known, or can change over time, or can change by any kind of property. For example, what I would like to stress today is the solvatochromic proofs to study membrane dynamics, for instance. This is not possible because we don't know what is the expectation. So we need to come up with a different approach. And this is where the phasor comes, the spectral phasor comes. And I will try to highlight with my example how this can be really a powerful tool to understand complex photophysics, still with a very simple approach, as Enrico said today. Very intuitive, very simple in rules. So the formalities are the same of the lifetime. So we Fourier transform our spectra, and we obtain, the G and S, which is the real and imaginary part of the transformation. And if we go to the formula, basically what we have is the sum or the integral of the intensity for each of the steps that we collect, multiplied by the cosine sine of the relative shift from the point of where your spectra starts. So channel 1 minus channel 1, channel 2 minus channel 2, and normalized by the total range. So we define what are the 2 pi that we have in the 4-quadrant phase. And then, as Enrico mentioned, in our phasor transformation, we always normalize to the total intensity that we collect. And this is important, and this is very important because it's what warrants in our transformation that all the linear combination and addition, etc., it works. Otherwise, you cannot claim for this properties. And this property will be very, very important for the analysis that we would like to have. So I would like to highlight that it's a model free, as Enrico mentioned, does not require previous knowledge. So just how the data on the phasor will tell you what you have and how to obtain information from there. It absolutely the traditional spectral analysis with the phasor is absolutely doable in Cuvette and an imaging. So you can do a single point, or you can do many single points, which are the end of the end is an image, right? So for an image, each pixel is a Cuvette. So, and it is very useful when we want to work with environmental sensitive groups. And I will try to convince you with the result that I picked for the presentation. Okay, so but how it works? So here to demonstrate how it works, I basically obtain, I simulate 3 Gaussian curves that it has a maximum that is shift, and it broaden over the shift of the maximum, right? So they have different full width at the HAL maximum. So for the first one, we will get a position, which is here with the arrow, and the same is for the red, and the same for the green, right? So, sorry, so for the green and the red. Well, if you look over, you can immediately tell that the position will appear basically counterclockwise, starting from this point here, 1, 0. And as farther you go, redder is the spectrum. The other information that you can immediately see is that as closer to the center you are, right, broader is the spectra. So we don't need any fitting or any analysis. We just can watch at the phasor and tell what is happening. When you have a point, it's very, very easy. But in an image, you can have easily millions of pixels. So this will be very helpful. The other important properties are the linear combination. So if we have any combination between blue or green, or blue and red, any other, it will line on line that join these 2 points. And of course, this is true for 3 components. But more importantly, and this is really relevant for images, if you have an image, you would like to know where your data is, because you cannot go pixel by pixel watching at your spectra, right? Or at your lifetime. So the reciprocity principle of the Fourier transformation enables you that if you use cursor to these circles, in your image, what are those pixels? And I generate here a 2x2 image to demonstrate this. This is the way that we can go in one case, so do the transformation of the phasor, but then came back from the phasor to the image. And it's also true that you can go and do a selection in your image and do a secondary phasor just for this image. And this is very useful because you can go pick back and forth, you know, trying to figure it out how you can do this. Well, this is another way of doing. So this is afternoon about Agdan, but you can see that there are an spectra on this image, and then you collect pixel by pixel the spectra, and then you calculate your phasor, and then you obtain, you know, use a cursor selection here from purple to red, and you highlight with the color that correspond to the different linear combination. What you obtain is rather an homogeneous image as you have here, a very heterogeneous image, which shows that there are some of these organelles that are purple, which means that are blue in the spectra, and the others are very green, very red, which are basically more red in the spectra. You see that from here to here, what you are changing is the phase, right? So you are immediately capturing the spectral shift. And you see that they are all in a linear combination, and we will go this in detail with the example that I picked. But we need to take care when we do these things, because, you know, one of the important things for this kind of imaging is you need to avoid saturation, because if you have saturation, of course, you cannot do a proper transformation. And here I just did an acquisition in order to show that you can do the transformation, of course, and I put just cursor here in order to see how it changed, the coloring in the image. This is no saturated, and as soon as I begin to saturation, you see that the pixels are moving from where it were before in the image, and then you start to highlight different regions. So saturation is an important issue for hyperspectral image. But also, you can think that eventually will be useful to having increasing number of channels in order to get a better phasor resolution. And this isn't true. And I will try to convince you for why. Well, you can acquire, for example, less number of channels in order to go fast. Well, this is good, but you will compromise your phase. By default, most of the truly hyperspectral detectors in the market are 32 channels. And this is sort of ideal situation because if you look at the distribution of the cloud and the phasor, this is the less broad, so the most concentrated phasor. So the signal to noise is sort of the best, as Nico was discussing before. If we increase the number of channels, well, first of all, we will increase the time because we will spend more time. In the 32 channel, we will get all the channels in one single shot. In the other, we will need 2 steps. And if you go farther, you will need more time. But the other point is, in each of the channels, you will get higher noise because you will get less number of photons per window. So signal to noise will optimize. And you can immediately see this in the phasor. The phasor is broadened. So you will not take advantage. And there is another important thing. Regardless of discrete, detector of 32 channels, when we transform the data to the phasor space, basically what we are transforming is from discrete function to a continuous function. So in the phasor, we can obtain resolution of nanometer in the center of mass shift. So this is another important thing. So the last thing that I want to mention is, well, the other problem of, if you want to go with high resolution or go fast and chop the phasor, so the total hyperspectral collection is that if you chop one side or the other your data, then you will not be able to claim for the linear combination properties. So this is to explain one day to someone what could happen with this. So this is night red in 2 different environments, triacylglycerol or phospholipids. And you for the phospholipids. You add the 2 spectra, you get this, and basically you can do the transformation. If you transformation, of course, you get everything in the line. You do the previous one, where you have the 2 points, pure points, and then the linear combination. You see that the point is outside of linear combination because you broke the linear combination by not having the total decay of the spectra, and this is really, really important. So now I'm going to explain a little bit about to highlight how phasor. I will talk really about these probes later on in the afternoon talk, but I want to at least give you, some explanation. So these DAM probes, dimethylaminonaphthalene probes, were synthesized by Gregorio Weber and Fred Ferris with intention to capture, environmental changes around the probe. And in particular, it was designed to study, protein dynamics, membrane dynamics. So basically, if to the dimethylaminonaphthalene, we include here in the R1, different group, we can give different affinities for different environments in proteins, membrane, or intracellular. The exquisite photophysics of this enable you to split about different things. So if you excite after excitation, the dimethylamino-naphthalene improves, so increase the dipole moment, and this dipole moment can be used to sense the environment. And how this can interact with the environment? Well, if there is no molecules that can be relaxed, can be reoriented by this dipole, basically it will fluoresce and it will sense basically polarity around the probe. If you have molecules that can be relaxed, for instance water inside the cell, some of the energy of this dipole in the excited state will be used to reorient the dipole active molecule around it, and it will reduce, the energy of the dipole and will emit farther in the green or red. And this can be used to sense what is going on in the environment of, proteins as well as lipids. So both parameters, lifetime and spectra, are modified by, these two processes, polarity as dipolar relaxation. Using lifetime, we can split both, and I will explain this today afternoon. With the spectra, we have the two together, because when we shift the spectra, is in regards of dipolar relaxation, but always of polarity. With the lifetime, if we select a particular region of the fluorescence, we can capture only the polarity or the dipolar relaxation. So in the case of Laurdan, the interesting thing is that the Laurdan is embedded in membranes by the Lauriga group that give you a strong traction by polarity, by the hydrophobicity. And we know since Tiziana Parassasi that basically it's able to capture a few molecules in the interface, which has restricted mobility, so it's confined water. And these molecules are basically, these molecules of water are basically modified in number by the physical state of the membrane, right? So if we go from, the membrane can be in different physical state, can be, disorder, can be shell phase, or if we add cholesterol, basically we can have a new membrane, which is liquid order. And Laurdan is able to capture this little difference in polarity as well in dipolar relaxation. And we will use this in order to study our system in biology. So basically, here is the spectra of Laurdan in membranes as the membrane is melted with higher temperature, and you see that the maximum is changing from 440 mainly to 445, 490 more or less. Traditionally, for this analysis, it was proposed to use this function, which is the generalized polarization function. And in this approximation, we are not using polarization. We are using just two channels, two bands of the fluorescence. So 440 and 490. And the reason for why this is important, the polarization function is important here, is because we have in the denominator the total intensity, and this enables you to have additivity properties of the polarization. So we can then quantify different fraction of one or the other. If you don't do that, if you do that radiometric, for instance, you cannot do this because these properties will not be kept. Later on in the time, basically, Luis Bagatoli, and I will declare more in detail about the history of this later afternoon, introduced the idea of use imaging, because we can collect with bandpass filter a region which is more assigned to the, in that case, more order membrane, and in that case, a more fluid membrane. And if we do this pixel by pixel, we can capture in a GUV, right, giant unilamellar vesicle, the fluidity of the membrane. And if you decrease the fluidity, you see that it's changing. And if we do this calculation and you use a color scheme, you can see very easily this transition in the membrane. And you can see that you start with 4 fluid, and then you have coexistence of 2 different membrane, and then you melt it back to more order membrane. one limitation that we have with the GP is that basically it's true that you can capture the GP of the shell and GP of the liquid, and then you can try to, untangle what is the fraction of each of the other in a complex situation. But the problem is this is not true. It's not full true, because when you have one membrane or the other, they influence each other. And these originally steps are no longer true or valuable. So you cannot use this as an input in the formula in order to obtain. So basically, this is a limitation, and this is important also in cell because you don't have these original steps. So you cannot say what is the beginning and what is the end of the GP in order to obtain the fraction. So let's see if with the spectral phasor we can do something better. So the first example that I pick is a simple example in cuvette. So you have DOPC. So DOPC is diolioyl phosphatidyl which is fluid at the temperature of experiment. DPPC is dipalmitoyl phosphatidyl which is solid at the temperature of the experiment. And if you put together 1,1, it will get a fluid shell coexistent. And if you increase the concentration of cholesterol, it will change from fluid, shell, liquid order, and then fluid, liquid order, and finally liquid order. If you capture the spectra and then you transform it to the GP, you see that you get some kind of trend of increasing GP by the liquid order occurrence, so increasing cholesterol. Well, if you want to understand, perhaps you need to propose a model. And a model can be very complicated, and you need to have physical explanation for the model. Well, let's see if phasors help us in that way. And basically, when we transform the same data to the spectral phasor, you see that it's very simple, that everything falls in a line, and what is changing is basically the fluidity and the dipolar relaxation and polarity, and all the points are in a linear combination between the beginning and the end of the trajectory. So phasors immediately show you what is going on in the situation. You have many different components, but all line between the origin and the end, right, of the trajectory. But what happens if you have a more complicated system? And this is a very complicated system because it is a pulmonary surfactant, which is basically the substance that we have in the lung in order to decrease the surface tension and enable us to breathe without collapsing the alveolus. And it's composed of 100 lipids plus 4 proteins. Basically, it's really, really complex. And if you do the phase transition, you see that the GP is changing. And you can immediately eventually tell that you go for higher order and then more fluid in this situation. But let's see if this behaves as a linear combination between two components or something else using the phasor. Well, when we look at the phasor, it show us that basically we are going from order to more fluid, and this is really not a linear combination of two components. We have, some kind of a bending of the trajectory, which implies that the Laurdan is exposed to a different environment, and we need to figure it out. Why? But in any case, it will immediately tell you that you cannot tell this is a two-component system. It's more complicated than that. Okay. The other example is on images, and these are the lamellar bodies. The lamellar bodies are basically the organelle responsible to accumulate the pulmonary surfactant in the epithelial cell, the pneumocystis type 2 in the lung. And what we know more or less about this organelle is by the electron microscopy. So we know that it has different steps in the maduration before it's secreted to the alveolar interface, where these lamellar bodies basically exploit, and basically they transform it in a monolayer that cover the epithelial lining fluid and the alveolus. And it wasn't that at that point when I was doing my PhD thesis, it wasn't that clear what was the organization of this lamellar body inside the cell in terms of the lipid organization. When we were doing the experiment, this other group from Spain showed that using the GP, they propose that these lamellar bodies inside the cell are basically on the shell organization, which means that are really, really packed. And at that point, it was very frustrating for us because we have almost ready the paper to be submitted, and this come out and basically struggle our publication. But lucky for us, we got phasors, and we were doing spectral phasor, and I will show you what phasor tell us extra of this information that enable us to make the publication. So what we did is we use a particular type of cell. These are immortalized cells that produce organelles, these lamellar bodies, and we culture over the time. And then you can see that the organelles increase in size over the time post confluency, and we capture the distribution. The distribution is heterogenic, it's not really discrete. And then we try to analyze how was the organization. So we transformate the Laurdan information into spectral phasor, and using the cursor selection, we highlight where are our lamellar bodies, and we can immediately see that the lamellar bodies at the beginning are more red, and then are getting more and more blue, which means that they are more organized in membrane, and they also grow in size. So the first question that we have is, okay, well, of course, if they grow in size, we will have higher number of pixels, which are blue, right? But can we correlate size and lipid organization? Maybe we can capture something else. What we discover is that, well, if we do that and we correlate size with the fractional number of pixels in the blue as well as in the green, I didn't say, but the blue is basically where the lamellar bodies are located. The red are basically fluid membrane, and the green are a linear combination between blue and green pixel, right? So different fraction of the two components. So basically, when we do that, what we discover is from day 1 to 7, they didn't change much in the fraction of blue pixel as well in the green pixel, but they grow inside quite a lot. But after that, they didn't grow much inside, but they really changed a lot in the number of blue pixels, telling us that at some point they completely shift the lipid organization. So how this can be? Well, we don't know, but we propose an explanation of that. Well, at the beginning, we got basically a preliminary version of the lamellar bodies. They are called multivesicular vesicle or composite bodies, that they are growing in size. And after some point, they are beginning to fill with the lipids and the protein. And after we reach that point, they got mature and they are organized in high packet organelles. What we discovered also is that this time information was very important, and in the next slide, I think on the other, I will explain why time information is important. The other important thing to answer was, okay, but these lipids inside the organelle, they are shell, they are liquid order, what kind of organization they have? Well, if we can prepare vesicles with Laurdan in shell condition, DPPC, or in a fluid condition, DOPC, or in a liquid order condition with a concentration of cholesterol like double of the one that we will have in the lamellar bodies, we got this position. And you see that if we have any pixel from the cell in this linear combination, we can tell that we have shell or fluid. And if we have something from the fluid to the liquid order, it will be basically a linear combination between one and the other. When basically the cells, the membrane cells, fall in the linear combination of the liquid order fluid, but the I549 lamellar bodies are a part of this trajectory. And how this can be? Well, what we discovered is that if we secrete basically our lamellar bodies and measure again in vitro, they shifted back to the liquid order, liquid disorder membrane, telling us that the organization of the lamellar bodies inside the cell is different of a liquid order, liquid disorder. And what we propose is that eventually inside the cell, due to the really high crowding condition, we have other kind of lipid organization because crowding influences the organization of membrane in producing non-lamellar structures. Okay, so I want to go further because I think I have my last 5 minutes. And basically, what I want to tell is a story that came up also in this paper, which is basically if you have, you know, the reviewer asked if we can demonstrate identity of these lamellar bodies in vitro. And basically, I got a plasmid with a protein who is only expressed in this organelle, so it's an molecular identity of the organelle. But the problem for me was this was labeled with green fluorescent protein, and at that point I was not able to do any transformation of molecular biology. So I tried to tackle this with biophysics and spectral phasor. So basically, the problem was green fluorescent it lie on the phasor here, and basically the Laurent trajectory is this. And you see that if you want to combine green fluorescent with Laurent, it will not give you really much information of what are, you know, the combination between fluid or solid with the green fluorescent. But the point is, well, we can use a different fluorophore, which is night red, who behave more or less same as the Laurent. And basically, if we do a co-staining of Laurent and night red, what we can get is basically the linear combination of, you know, Laurent under relaxation and night red under relaxation, and Laurent under, no relaxation, Laurent on relaxation, and night red on relaxation. And this is immediately, you see the phasor that the linear combination show up very, very, you know, easily here and here. And we can color and we can identify our organelles. So now we can use night red as a label for the membrane, and we can use, you know, our green fluorescent proteins as an identity. And when we do that, you see that in the purple here are the linear combination between night red and the night red, and the night red and the green fluorescent. And these are our lamellar bodies here in purple. And then we can do all the other linear combination in the cell for this experiment. So basically, we demonstrate that this is possible, and basically the identity of the lipid. To finalize my talk, I want to highlight this hyperspectral imaging in scattering media and without using a hyperspectral detector. And I think Sasha Dvornikov is going to cover this in detail, but what I want to tell is, well, when we do phasor transformation, we do math because we collect the spectra and then we apply the phasor transformation and then we get the phasor. But what happens if we have a filter with a shape of transmission, we refer to a sine and a cosine function? If this is possible, it is possible, well, we can obtain the phasor without obtaining the spectra, right? And this is what Sasha Dvornikov and Enrico proposed in this paper. And you can see that if we do that and we do the proper calibration of mathematics, we can capture, these are beads with two colors, and you can see that you can capture the green, which is, you know, less modulation, and the orange, which is high modulation, higher phase. And of course, if you look carefully here, you see some little dots which are the pixel on the frontiers between one bead and the other, which are the linear combination. And also you can do this for many other applications, like, you know, collagen characterization in the cell. This is a bond from a mice, and you can acquire this in lifetime, but also you can acquire spectra and you can get exactly the same information due to this approach. So in the last minute, what I want to tell is how we can expand this opportunity of sine and cosine filter for camera base. So one problem with the camera base on the camera base is, well, either you have to, if you want to scan spectra, either you want to renounce to time, so you acquire spectra pixel by pixel, or you are acquiring spatial information X and Y and then spectra at rather time. And this is compromises of time. With this approach, we can collect almost in parallel 3 channels, so cyan, cosine, and total intensity, and then do the transformation and capture the phasor in a camera, in all the pixels of the camera. This is really, really an opportunity because we can do really fast thing with the spin configuration, the light sheet configuration. For instance, we use this to capture, the different lineage of cells in the retina. This is a particularly model in zebrafish that express, different fluorescent marker for different kind of cell under development. And basically, what we did is take an image and obtain the spectra through the cyan cosine filter. You can see that basically the overlapping of the fluorescent are very, very high, which really compromise the possibility of, splitting by channels. And to complicate our life, we also put an extra marker, which is a marker for the nucleus. So when we obtain the data and we transform the phasor, we immediately can see that the phasor can be split like in 2 linear combination, green, yellow, cyan, and magenta here. And if we highlight the retina, you can more or less recompose all the different components for the different cellular markers. And if we do a plus, which is, well, isolate the region of interest in the image and look at the linear combination, what we discover is that we recover exactly the linear combination that are expected for the different regions. So when you have, I don't know, cyan and green, means that you have this protein or this cell. When you have this other, this other. And this is really the power of the phasor in order to understand very complicated things. And I want to highlight that here we have only one little spot, which is here in purple. And these are this pure amount of pixels here, and the phasor capture very, very well that these are different locations, which is really complicated in the convolution because you are going to lose if you have 5% of component, you will going to lose. So I don't have time to go there. I would like to highlight some new approaches using phasor. So Enrico already mentioned about Lorenzo talk tomorrow in truly high spectral film. Luca Lanzano will talk tomorrow about how phasor will be useful for super resolution. Belen will talk about a new way to do, phasor with instead of cyan cosine filter, up and down filters. And there are some other applications for a different kind of microscopy, but very, very useful for in vivo imaging. So thank you very much.